Hello, everyone. Wanted to welcome you to San Diego's multifaceted attack against COVID-19 virtual community event. We had met back in June, and we're bringing some of our speakers that spoke previously to actually talk about the updates that they've made from since June of 2020. We have Joe Payne, who's the CEO and founder of Arcturus Therapeutics. We also have Susan Tousi, who is the Chief Product Officer from Illumina. And we also have Sonia Sharma, who's the Director for the Center for Functional Genomics at the La Jolla Institute for Immunology. Thank you so much for being with us today. And Joe, I want to go ahead and start with you and to see if you can tell us about the progress that you've been making with your mRNA COVID-19 vaccine, both here in the US as well as in Singapore.
Great. Well, thanks for the introduction, Pam.
You're welcome. Thanks for being here.
Yeah. Well, since June 2020, a lot's happened. We've initiated a phase 1/2 trial in Singapore in over 100 folks there and established some a nice proof of concept and some initial safety and tolerability data with the vaccine. We've transitioned to a full phase 2 study here in the US and in Singapore. In fact, we have an ongoing, we have a site that recruited patients that were dosed in Coronado Island, right nearby. So we're vaccinating people here in the US in form of a phase 2 trial, and that's going well. And so yesterday we provided a quarterly update. And so where we are is we have a single shot vaccine. Everyone's familiar with the I guess the Moderna and the Pfizer messenger RNA vaccines, but the Arcturus vaccine is a single shot messenger RNA vaccine instead of a prime boost mRNA vaccine. And yesterday we announced that we're planning to initiate our phase 3 study in Q2, which is just around the corner. It's going to be involving several countries, of course, in areas where there's a high prevalence of COVID, in areas where it's easy to recruit for a placebo-controlled trial. There's over 200 countries in this world, and the strong majority of them haven't seen a vaccine yet. So there's a high sense of urgency for us to run a clinical trial that's a placebo-controlled trial, meaning we'll be able to get that efficacy number for our vaccine as well, hopefully soon. But a lot's happened there, and we are a messenger RNA medicines and vaccines company at Arcturus, and there's more to messenger RNA than just vaccines. mRNA came of age in 2020, and so people are asking, what else can mRNA do, this messenger RNA technology? So we have an exciting liver franchise and a lung franchise that we're building as well. So since we've last talked, we've injected messenger RNA intravenously and very safely in healthy volunteers, and shown that our technology is safe with intravenous or systemic administrations of RNA. And we have another program where you inhale the messenger RNA for cystic fibrosis that's matured into a development candidate that we've announced since we last met. So a lot of activity, no doubt. We're excited. The company's growing. We look forward to continue to grow right here in San Diego. I'll turn the time back to you.
Okay, thanks, Joe, and congratulations on your success. I know you've raised a lot of funding, and there's been tons and tons of activities, and I've known you since the beginning when you first started Arcturus, and it's great to see your progress. So thank you. Thank you.
It's good to be here.
Thank you. So Susan, when we last spoke back in June of 2020 at our last event, Illumina had just received the FDA emergency use authorization for your sequence-based COVID-19 diagnostic test. And I wanted to see if you could tell us more about the advancements in the help against the fight against COVID-19. I know you've made tremendous progress with that.
Yeah, absolutely. And yeah, it's great being here again. with all of you, and Jolla and Sonia as well. We were kind of pulled into the fight for COVID. We had not been on our radar in terms of our product roadmap. But since our technology was used we were called into Wuhan in December 2019, and the first genome of the virus that was sequenced and then kind of published, and that really started the clock on much of the vaccine development and therapy development. And so we knew we had a role to play. We kind of pulled it together pretty quickly to get emergency use authorization on end-to-end COVID-seq test, and this is a test that sequences the virus. It runs 3,072 samples and kind of a 24-hour turnaround on our NovaSeq 6000 platform. Well, since then, we've really kind of grown our entire offering around this. We have RUO-based COVID tests across all of our platforms, our mid-throughput and low-throughput platforms. We have a rapid run test on our MiniSeq platform. We've published a number of a whole data toolkit, which includes apps that run, a metagenomics app, a COVID-seq detection app, a lineage app, and we've seen just tremendous uptake. At first, we knew that next generation sequencing might not be a frontline test like a PCR test, but that it had a role to play, and that tracking mutations of the virus, being able to publish and share those, and really seeing how the virus was going to evolve and eventually kind of surveillance would be incredibly important. And here we stand now talking about the variants of the virus, and that being our next concern. So we have had tremendous uptake. In fact, we serve 10,000 labs, 115 countries. We're seeing the interest in surveillance growing. So just as I was mentioning to Pamela before we started, that in the U.S. We saw a 70-fold increase in the number of positive samples that are being sequenced, and this is through the CDC and our work with the CDC. We are supporting efforts at the CDC, the Chinese CDC, the NIH, the NHS in England, partners and collaborators like Broad and Stanford and Gates Foundation. We've put sequencers into Africa, and we also have released a number of panels that are for detection of a large range of pathogens and also microbial kind of resistance markers. So we're just seeing a tremendous interest and increase in all of those offerings. So we know that we're serving a need here, and certainly the uniqueness of our sequencing technology to be able to capture the entire genetics of the virus and its evolution, and do that kind of at scale and do that very fast is incredibly important. Our data tools for kind of sharing that, being able to publish to the GISAID or other kind of public databases is also incredibly important. So we've been happy that we've been able to play a role.
Awesome. Thank you so much, Susan, for the tremendous progress that you've been making. And I know you lead a team of 1,400 people that are global, which is an amazing task in itself. And I mean, how has it been managing the team in the pandemic?
Yeah, it's been a real challenge, but I'm just so pleased to see how our Illumina employees kind of rise to the challenge. And we have, for me, for the people who kind of develop our products and have do a lot of work in labs, they need to be on site. So I've had about 30% of my team that is kind of regularly on site throughout this time, through developing the COVID-seq test, through developing our oncology test, our non-invasive prenatal testing kits. And, as we saw some of the other kind of diagnostic tests decline during the worst of the pandemic, we're seeing that all now come back, which is great to see. Obviously, we want to make sure that people are being tested for pregnancy health, for oncology. And it's, our people are incredibly motivated. We've put in safety measures in all areas. We've been incredibly disciplined. We have our own testing facilities, of course, so we run our own testing, and we practice social distancing. People are wearing masks, and we've had very kind of safe work environment, which we've been happy to see. I was talking to one of our commercial team actually, who they described like having to have been tested 4 times to go into Estonia to, serve a customer. And, they were tested twice as kind of like they got on the plane, they got off the plane, they're getting to Estonia on the customer side. So people have jumped through like many hoops, but through all that, it's been really a pleasure to see how we've been able to serve customers, and actually our customer kind of positive response rate has been very high. So it's a challenge, but we rose to it, which is great to see.
Well, awesome. Thank you so much for sharing that. Okay, Sonia, can you tell us more about your lab's work to study inflammation and immunity in the context of severe COVID cases in specific ethnic groups?
Absolutely, Pamela. Thank you so much. So just to give you a little bit of background, my academic, so I run an academic lab here at the La Jolla Institute for Allergy and Immunology. And so just to give you a bit of background, our institute is a nonprofit, basic, and also translational research institute that really solely specializes in studying the immune system and immune-related diseases. What are these diseases? Virus infection like SARS-CoV-2, which causes COVID, cancer, autoimmunity. My lab in particular at LJI, and we've been active for 8 years, since 2013. We use primary discovery-based approaches, including, transcriptomics, proteomics, and others, to really pinpoint the molecular drivers of protective immune responses, as we would see in antiviral immunity, for example, to SARS-CoV-2, but also pathogenic immunity. And the example of this would be COVID-induced inflammation. Our lab in particular, as I said, we're pretty translational. We use a humans first approach to identify relevant molecules using omics techniques that basically are profiling DNA, RNA, protein, and more recently, small molecule metabolites in patients. And so again, our research is very translational. So using this humans approach to study COVID, since June, and in the last year, and more in particular in the last 6 months, we've been applying a technique using mass spectrometry-based metabolomics to analyze, profile small molecule metabolites that are present in the blood of COVID patients. And why? Well, pretty much these small molecule metabolites that are in our blood, they really directly influence, they're really the effector molecules of protective and pathogenic immunity, as is observed, as is very relevant to COVID disease. Now, how would we apply this clinically? Well, these molecules that are circulating in our blood, they could be used clinically as biomarkers to predict the outcome of mild or severe COVID disease using a simple blood test, for example. Alternatively, some of these molecules are actually bona fide immune modulators and could be harnessed potentially as therapeutics to influence directly the outcome of COVID disease. So, as you mentioned, to make our studies as broadly relevant as possible, my lab is trying to study diverse patient populations. So focusing, for example, on hundreds of patients that we've amassed in the Los Angeles, San Diego region, which is ethnically diverse in terms of having significant amount of Caucasian, Black, and Latinx population. Because it's important to note that we expect the disease to share obviously broadly similar molecular characteristics, but also have distinct molecular underlying factors. And we know this just from the epidemiology. So I want to mention that our studies are all collaborative. We're working with several different researchers, for example, with Mohit Jain at UCSD for metabolomics and Dr. Susan Chang at Cedars-Sinai Medical Center for the human cohorts. So to give you like specific example of something we found. So we analyzed blood samples of patients in the LA area. And again, these are patients at Cedars-Sinai Medical Center who are already under the care of physicians there, or they could be the healthcare workers who are also part of the system. So what we found is that in patients who developed severe COVID, they were compared to patients who had mild COVID. The patients who developed this severe disease were actually missing a key metabolite in their blood that was actually present in the blood of patients who had mild disease. And this is just in itself a pretty simple, like fundamental observation, but leads to a lot of questions in that the real work begins now. I just want to mention, this is a very kind of cutting edge experimental technology that we're only really beginning to understand, the potential of. But the real work begins. What is this potentially protective molecule? What's its chemistry? How does it work to potentially boost or suppress the immune system? Is it conserved in males and females and in our different ethnic populations within the LA group? Is this molecule present in children compared to adults? And maybe most importantly, does this molecule appear in the blood of patients with mild disease early enough that could be used as a biomarker to specifically stratify patients who won't need a ventilator or maybe won't need remdesivir or another treatment that could be saved for a patient who we would expect to develop severe disease. So that's the potential power of the technique, and we think it could be a game changer in how we treat COVID.
Wow, that's amazing, all the progress you're making, and just also the importance of a collaboration and really looking at the full diverse ethnic group and how this disease affects people individually, which is tremendous. So thank you so much. And Joe, I know you spoke about one of the ways that Arcturus's vaccine was different is that it was a one-dose shot compared to Pfizer and Moderna. Wanted to see if you can tell us what some of the other unique differences are, and then also if you had an estimated timeline on when you would hope to have your clinical trials completed.
Sure. Great question. So the Arcturus vaccine is a next generation messenger RNA vaccine. It's not a conventional messenger RNA molecule. It's a technology called self-amplifying messenger RNA. So the reason why this is a single dose is because when a conventional messenger RNA enters a cell, it makes a bunch of protein. It makes an antigen, the full-length spike protein that causes the vaccine response. But it makes it for a couple days, but then the messenger RNA molecule degrades and it requires a second shot. The self-amplifying messenger RNA is different. This is the Arcturus vaccine, and it's the next generation technology of mRNA vaccines. And this self-amplifying mRNA, when it enters a cell, so after you get that injection in your arm, it makes the antigen, the same antigen or a similar antigen, but it does for an extended duration of time. And because of that, there's a lot of benefits with this technology. Number one, the dose level is a lot lower. Our dose level is 5 micrograms, not 100 micrograms like the Moderna vaccine times 2, so 200 micrograms. So that brings potential safety advantages. Because you're injecting less material, less RNA, less lipid excipients and stuff like that. So it could have safety benefits, but also manufacturing benefits. You can appreciate that, one batch of Arcturus messenger RNA vaccine, because of the dose level, could be many other, manufacturing runs for other vaccines at higher dose levels. So we can treat a larger portion of the population more quickly. With respect to the other vaccines are all distributed as frozen liquids. Some need to be stored at -70 centigrade, and this presents some challenges when you're distributing a frozen liquid, because if it melts, you have to use it or you have to discard it. Where the Arcturus vaccine is a lyophilized version of the vaccine. So it's freeze-dried. So there's no water in it. So it's more easily distributed, and that's another advantage to it. So not only the low dose level, but it's lyophilized. And then finally, we just shared data yesterday that came out of the Duke NUS Medical School that our cellular immunity after a single administration was 8 to 10-fold higher than conventional messenger RNA. Now, why is that important? Is these T cells or the cellular immunity, they're very helpful for protection, but also durability and variant coverage. So everybody wants a vaccine that's protective, but why not have one that lasts a little longer? And why not have one that provides coverage or protection against these new variants? The COVID-19 is changing, and or mutations or variants, they're called. And some of these changes or versions of COVID can be more dangerous, more contagious, more of a challenge. And with cellular immunity, these T cells may have broader variant coverage relative to antibodies, or what's called humoral immunity. So we're bringing forward a different type of messenger RNA that allows to lower the dose level, allows us to distribute this as a lyophilized vaccine, and it improves the cellular immunity, which could improve durability and variant coverage. So it's an exciting technology that we're proving out in phase 3 studies upcoming here in the second quarter. You ask about time. When is this vaccine going to be available? Because the world definitely needs single-shot vaccines. Just ask J&J. They just approved one of the first ones, and there's extraordinary demand for a single-dose vaccine. We're talking to the leaders of the world on a regular basis. In fact, right prior to this call, but there's extraordinary demand for a single-shot vaccine because of the simplicity, the convenience, and compliance associated with it, and the reduced cost of distributing. You can appreciate the logistical challenges for many of the countries in this world to distribute a 2-shot vaccine. Hurting humans is harder than hurting cats, okay, for that second shot. So it's difficult. But when are we going to get results? Well, you saw, 6 months ago, there was plenty of COVID to provide exposure to the placebo group. And that's what is key. But this year, there's even more COVID in many of these countries. So the prevalence is higher. So the clinical trial may not be as long. It could be just a few months, and we could have our first readout. And we're planning on getting emergency use approval in the second half of this year, depending on when those read out. And once you have emergency use approval, like you've seen here in the United States, then we can proceed to distribute the dose. We've already initiated stockpiling. And again, because of our manufacturing process and our dose level, we're well ahead of the curve there. So we'll be able to distribute doses to the countries that have already engaged us for supply agreements. Singapore has entered into a contract with us, Israel as well, and we're in discussions with many countries with respect to supply agreements. So we hope to be able to make a dent this year, and then going on to in the future. All of the vaccines are no longer. It's no longer going to be a 2-shot experience going forward. It's all the. We fully expect it that the market will be a single administration market, whether it's a booster shot or for people that have never received the vaccine. It'll be a single dose vaccine for next year. So that's what we're aiming to help. And then the final comment here is there's a subtype of vaccines called viral vector vaccines, and great companies like J&J and AstraZeneca, countries like Russia, India, and China, they all have viral vector vaccines that are being distributed. That means they were utilizing a virus that delivers the vaccine, not a lipid nanoparticle or other things that other vaccine companies like Arcturus are using. So these viral vector vaccines may have increased challenges with respect to redosability next year because of their delivery system. So we'll, Arcturus and many other companies will be playing a role in the endemic phase next year. We believe that we'll be helping these people transition from the viral vector vaccines to something that's redosable on an annual basis.
Okay, wonderful, Joe. So it sounds like wishing you luck. There's lots of advantages. It's one dose. You're giving only 5 micrograms. In addition, when it's delivered in a powder, so you don't have all of the temperature issues and other things. And it sounds like with the variants, you feel like you'll be able to cover the various different variants based on the mRNA technology.
Yeah, if I can interject there just to explain it a little bit further, because many people here may not appreciate this, but some of the common colds are coronaviruses. And if someone gets a common cold, it's a coronavirus, and your body builds up T cells, they're called, cellular immunity. And these T cells, even though it was built against the common cold, they can be protective to COVID-19, a completely different pathogen within the coronavirus family.
Mm-hmm.
So that's not a variant. That's a completely different pathogen. That's how broad T cells can protect. So if we generate T cells against COVID-19, we may be able to protect against a wider list or of all the different versions of COVID-19. That's what we're hoping. That's what the theory is suggesting. So, we're very happy to see that we have a robust T cell response to our vaccine, and that hopefully will have an impact on variant coverage. But if it doesn't, if there's some crazy new variant that comes up that escapes the protection of the Arcturus vaccine, it's okay, because we're still an mRNA vaccine, which means it's readily updatable. We can rapidly change that payload, like in weeks, it's not months. So we could simply roll that out. And just like influenza, the experience that people have, they go in every year and they update the vaccine on a periodic basis. That may ultimately be what happens for COVID as we return to some sort of normalcy in the coming years, is a periodic, either annual or every other year vaccine for COVID that's readily updatable. But plan A, just to be clear, is let's hope that the T cells are broadly variant covering. That makes it a lot simpler for us, and it's a more commercially attractive it'll be a more successful product if it has broad variant coverage. So that's what we're hoping for.
Okay, great. And you know what, real quickly, Joe, a question just came in from Nushin is, how long are you expecting the vaccine to be effective? Do you have an answer for that yet?
Yeah, well, we shared data yesterday, or the Duke NUS Medical School did, where they showed that our T cell responses were 8 to 10-fold higher than conventional mRNA. That could have an impact on durability. T cells themselves have about a half-life of 4 months. Antibodies have a half-life of 3 weeks. So there's about 6-fold difference. So T cells last 6 times longer than antibodies. But that is an oversimplification. Any immunologist will hear that it's more complicated than that. That was a long way of saying we have a theoretical rationale for a longer, more durable vaccine, but we have to prove it. The humoral immunity these antibodies, they can help impact memory cells and be longer lasting, even though the antibodies don't last very long. You hear about these stories where people don't have any antibodies, but they're still protected because those antibodies did their job and they got converted into memory, and they still have longer protection. T cells are just better at it. And T cells, they get formed, but they do what's called contraction. They come back down and they get converted into these memory cells. And these memory cells are embedded in your tissues, and they just stay there in your organs and your tissues waiting for the next exposure. And they're very protective, and they last a long time because they're hidden. They're hiding and just hanging out in your tissues for a long time. So hopefully it'll last a longer time. And to what degree, I can only comment that after a single injection that our T cells are 8 times to 10 times higher based upon the sub-small interim data that we've collected in humans. And hopefully we'll repeat or mature that data set with the 500 plus people in our phase 2 trial that's ongoing.
Okay, perfect. Thanks, Joe. That was really helpful. And Susan, so obviously we're seeing a number of mutations, variants to the original COVID-19 virus. What is Illumina's long-term plan to keep pace with these various mutations?
There's a lot to do, and I'll kind of start with my co-panelists here. We will continue to serve the research community, like Sonia talked about, in terms of really understanding the genomic markers of patients who suffer varying degrees of the disease and why. So there's a lot to understand in terms of immune repertoire or other genomic aspects of why some people get very sick and even die, and why others basically don't even recognize that they have it. We will continue to serve the vaccine developers, because I think tracking the mutations of the virus, knowing exactly how to evolve these vaccines and how to change the payload so these mRNA vaccines continue to be effective to mutations of the virus will be incredibly important. And then more broadly, I mean, we think that the opportunity, we have the technology and the opportunity for broad-based surveillance. I mean, the world has suffered from these diseases, these infectious diseases. We've had various forms of SARS that have, killed many people. We have had, Ebola and other, diseases. We're going to have other diseases that go from the animal population to the human population, and we shouldn't. We don't have to be in this position of a pandemic. We have the technology to be doing broad-based surveillance on a regular basis. And our CEO, Francis D'Souza, has put out this concept of a bioforce. And just as, some countries like the UK have a cyber force and we're talking about a space force, why not have a bioforce that is, constantly doing some level of surveillance of biohazards in our environment? And being much more prepared, we could start the clock on, the actions that we've taken to stop the spread of the disease. But then, of course, the therapy development and vaccine development much earlier. And I think, that we need all the countries and the public health to kind of rise to the occasion. There's got to be data sharing across the countries. We've seen that, there's no limit to how viruses can go from one geography to another in our very connected world. So there needs to be cooperation. But we have the technology, and we'll continue to drive our roadmap of the technology, making it more cost effective, making it kind of easier to adopt, and more scalable. And so we'll continue there, and we want to serve those who, as I mentioned, a number of our collaborators in public health globally, as well as our collaborators in the research community. We want to serve them to help put in place a really global surveillance system, a bioforce that will keep this from ever happening again. That's kind of how I see our long-term future, in addition to everything else we're doing around it.
Thank you so much, Susan. And yeah, the bioforce is so important. As we saw, we weren't prepared, and we really have to be thinking ahead. And so thank you for sharing that. And I saw Christina sent information on how to get more information about bioforce. So
yeah, we have an op-ed piece in The Economist that Francis wrote that we're glad to share to this community.
Okay, awesome. Yeah, if you send that to me, I can send it out to everybody that attended.
Absolutely.
Okay, so Sonia, so wanted to see if you could give us an update. Overall, La Jolla Institute of Immunology has been very active in the fight against COVID. In addition to that, you have the Coronavirus Immunotherapy Consortium, which is funded by the Gates Foundation. And I wanted to see if you could give us an update on the progress being made there. And sort of to tell the group a little bit more about what that's all about.
Yeah, I can absolutely speak to that in some part. So this consortium, which is a collaborative effort with the Gates Foundation and LJI, and it's specifically Erica Ullman-Sapphire at LJI. A part of this consortium is to bring together a group of academic labs in order to identify, in this particular case, study, the best neutralizing antibodies for CoV-2. Now, I just want to be clear. So, there are several. LJI has really, I'm very proud to say that, we've really been active in the basic and translational research with COVID. So Joe, you mentioned some work with memory T cells, and that work actually has been done by Alex Sete and Shane Crotty at our institute.
Mm-hmm.
The Gates Foundation has done has this collaborative effort with Erica Ullman-Sapphire. I would rather speak more specifically to my lab-specific collaborative project with Gates. And I just want to say this is a little bit distinct from the work of the Coronavirus Immunotherapy Consortium. But it is related in that we're all at LJI and we're all studying COVID and we're all doing basic research. So, and just to bring it back to what I said in my first part, as an emerging experimental technology to study COVID disease in COVID patients, we've been working with Gates on the development and application of metabolomics. And that's something that's actually in general of high interest to the Gates Foundation and is currently actively funded by the Gates Foundation. So just to put it in perspective, like any technology, this is an expensive technique in terms of machinery, in terms of mass spec, in terms of data analysis, computer server storage even. And the Gates Foundation has already committed to funding this research for academic labs with a particular emphasis on bringing these technologies, these emerging cutting-edge technologies to countries and ethnic groups and researchers who wouldn't already have access to them. And that's where it goes back to what we were talking about performing metabolomics in ethnic populations. And this is something where the Gates collaboration is coming in. So we're specifically working with the foundation to extend our ethnically diverse studies that I already talked about to specifically analyze blood samples from patients in Thailand, Nepal, and Vietnam to analyze, metabolites associated with severe COVID disease, as we talked about. Basically, these blood samples from the COVID patients at hospital centers in these 3 countries will be sampled, shipped to La Jolla, and analyzed using our metabolomics techniques to identify those novel molecules involved in either predicting or maybe modulating severe disease. And again, these international studies are also a collaborative effort. It's myself, Dr. Sujan Shrestha at LJI, and Dr. Meng-Ling Moy at Nagasaki University. And so we're really excited to do this collaborative work. It's really just starting. We're still in the developmental phase with the Gates Foundation, but they've really provided, much like with the Immunotherapy Consortium, they're really providing an opportunity for us to compare and contrast the biology that we're observing in the US patients as what with international patients.
Thank you so much, Sonia, for that, and for all the great work that you and your lab are doing, as well as everybody else at your institute. And so I wanted to open it up just to hear everybody's viewpoint on where do you see things going and heading overall in 2021, or in 2021 and then into 2022, and when do you see things heading back to somewhat of a normal? So I don't know who wants to go first on that question.
That's a tough one.
I can go first. Yeah, I'll
Okay.
The eternal optimist. I tend to be. I think that the new world, the new normal, in terms of kind of healthcare and our progressive response to what's happened is going to be much better. I mean, I feel that we're seeing momentum in our business, not just because of COVID, but because of kind of interest in molecular diagnostics in general, and much better targeted care, much simpler diagnostics to begin with. We're in the kind of quiet period of an acquisition of GRAIL, which is using liquid biopsy as an early detection of cancer, all the way back to kind of tissue of origin from a simple blood test, phase shifting cancer into where it's not a life-threatening disease. So we've seen, the uptake of telemedicine. We've seen the uptake of kind of more preventative medicine. We're seeing risk stratification, so people know when to come into the doctor and when if they don't need to as often. So I think we're going to see technology play its role, and certainly for us, we're super ambitious about our product roadmap and the role that we play, and we think that the usefulness of genomics, the usefulness of multiomics in early diagnosis, in much better kind of targeted therapy, in prevention of the spread of infectious disease, as we talked about with surveillance, is really unending. And I think that there is not only going to be a resurgence. It's great to see the new administration having a cabinet position that Eric Lander is serving as as a kind of scientific officer for the country. Our board member Francis Arnold, who's a Nobel Prize winner, and Eric both kind of co-chairing a technology committee. I think there's going to be a resurgence in kind of technology investment, scientific investment, and a much better future based on richer information.
Awesome. Thank you so much, Susan. Sonia, do you want to go next then?
Absolutely. I just want to speak to, I mean, I agree with everything that Susan said. And I just want to say, in talking about stepping back a bit, I'm cautiously optimistic that, the vaccine rollouts, not only, as we saw with what Joe was saying, not only the already approved vaccines, but the new vaccines, which will be better, quote unquote, what everything he talked about, lyophilized, easier to deliver, single shots. The idea was always that, with about, let's say, 100 potential vaccine companies, if one was successful, at least half of them were going to be successful. So, and I'm really optimistic. And I think that if the vaccine rollouts continue to progress and the production is ramped up and distribution gets better, and I'm optimistic about it, I really am, that we'll control the virus. And of course, emerging variants are the concern. And hopefully the immune system will be able to fight against them. But because of that, I'm just speaking more day to day. I do see us transitioning to somewhat normal life over the next year. And what I mean by that, if the vaccine rollout continues and progresses, I see schools and businesses opening and return. Although, can you put the toothpaste back in the tube? I mean, I think that precautions like working from home when possible, masking, some social distancing, I see that to continue to be a part of daily life, partly because of maybe the emerging variants. And I think testing is here to stay too, at least for the next year. But I'm optimistic.
Well, thank you. Thank you so much, Sonia. I think we all want to be optimistic for sure. So, Joe, what do you have to share?
Well, I think the comments that have been made are great and accurate. There's no reason to believe that the vaccines are going to have an initial wave of efficacy. We've already seen it. So I think the challenges or at least the discussions is how do we appropriately vaccinate our children, subgroups like pregnant women, increases on, with the increased choice of vaccines will become available. This is a great thing. It's a good problem, as people are going to try to understand, what's important to them. Is it durability or safety or there's political issues that are going to come forward, whether it's the rights to be vaccinated and what's mandated. You have to be vaccinated to attend a football game, or you have to be vaccinated to work here. This kind of stuff. So there's going to be a lot of politics about vaccinations. Political pressure externally as well. The U.S. will have copious amounts of vaccines and access to vaccines at some point. And I think at the latter half of this year, there's going to be a good political discussion of how we utilize these excess in vaccines appropriately to leverage them for political benefit or for humanitarian benefit. And it'll be an interesting political discussion. So I look forward to seeing how this plays out. I don't know how I'd label myself, if I'm an optimist, a pessimist, or a realist. It depends on who you ask and what the topic is and what day. But I think I think it's going to be a great year to establish some sort of normalcy. The caveats are, as long as people realize that COVID is here to stay, it's global, it's pathogenic, it's contagious, it's airborne, it's not going away. So there's going to be a lot of naivete that we have to work through, and just make sure that this is managed properly in the endemic setting. I forecast another wave coming back in, because people don't think they need to take the vaccines, or they're not respectfully considering the variants, but it won't be to anywhere the near extent that we've experienced in 2020. But there will be a little. I anticipate some little waves of COVID, and let's hope it's not a big one pertaining to a nasty variant that escapes the first moving vaccines. That's not anything that we'd wish for. I think I'll step off I'll step away from the mic and pass it back to you, Pam.
Yeah, no, thanks for sharing. I think we can all say, like, the amount of progress that we made in this past year is just tremendous. It's beyond what we could have ever expected, and going forward, I think we can be optimistic. There was one other question that came in from Kathleen Molnar, is on whether the speakers could comment on the prevalence of and the effect of the spread of asymptomatic cases.
I mean, just to talk a little bit, and again, I'm not an expert on this, but it's my impression that the majority or a significant portion of cases are asymptomatic, and that may be due to what Joe mentioned, the work by Sete and Crotty demonstrating that pre-infection with coronaviruses can potentially confer some degree of T cell protection. And it's thought that this may be contributing, this memory T cell response to coronaviruses may be conferring some protection, some disease progression protection, and allowing for these asymptomatic cases. Now, conversely, we've also heard that asymptomatic cases can cause spread, and that's what I know of it. I don't know what you folks think.
No, I have no further comment. I think that was a great answer.
Yeah, we'll see. I mean, it's so interesting to think that if the T cell responses are cross-reactive as we think they are, it would just be the best news. And let's hope.
Awesome. Thanks. Susan, do you have anything to add?
No, I agree with the comments. I also agree with the fact that we're just going to have to be vigilant for some time to come. And, until we have some assurance that there's not a mutation out there that's not going to get past these first vaccines, I think, and the fact that there is asymptomatic spread for people who haven't had the vaccine, or these who knows about the new mutations. We're going to have to be vigilant for some time to come. I kind of agree with what Sonia said, in that testing is here to stay for some time, and as is just kind of taking the precautions that we have. We can't let our guard down. Now, I don't think it will be anywhere near the pandemic that we had. And in the future, once again, I think in the future, we've got to use the tools and the technology that we have to kind of never be in this situation again.
And shifting the conversation a little bit when we're talking about the future, I think it goes without saying that when billions and billions of dollars is invested into a certain area, there's downstream effects that are going to be very, very, very positive going forward. Like the understanding the human body and the immune system, immunology is, vaccines were ignored for decades. People tried to raise money for this very, very important area of science for decades, and now this influx of capital. But it's not just for vaccinology, immunology, but in the mRNA space, all of this manufacturing and technology and improvements that we've experienced is going to be applicable to completely different diseases, all because of COVID. So this horrible 2020 is just, the silver lining here is it's going to impact everything from cancer to liver therapeutics, lung therapeutics. There's so many people are going to have just access to this technologies that have been accelerated by 10 years because of this pandemic. And then just the understanding of the science, like all the money that's become just available to understand all these immunological processes, right, is going to have not just near-term effects, but downstream effects. And I know that's kind of stating the obvious, but it'll be fun to see this, instead of a wave of COVID, a wave of innovation that we're going to see multiply into something special in 22 and 23. The impact of all this capital from the government to all these private institutions and globally is going to be great to watch.
See, now you are the optimist. Do you see your mRNA technology kind of applying to cancer?
Well, there's 5 mRNA companies that are publicly traded. The leading messenger RNA company, in my view, is BioNTech in cancer vaccines. So the fact that they're the BioNTech Pfizer vaccine is going to be successfully addressing a lot of this pandemic, all that capital, that commercial revenue is going to be applied to their cancer programs. I think that's great news for oncology, guys, that these messenger RNA cancer vaccines are, $15 billion of revenue is a lot of money. I'm not saying all of that's going to cancer, but it's going to help. And so I think there's applications. For messenger RNA, it's all about getting that messenger RNA molecule to where it needs to be. It's a fantastic holy grail therapeutic medicine, but getting this big, long, highly negatively charged molecule to where it needs to be safely and effectively is very difficult. There's 200 different cell types in the body, and Arcturus is good at 3 of them. We can safely and effectively deliver messenger RNA into myocytes or muscle cells in the arm for vaccine applications, and hepatocytes in the liver through an intravenous injection, and then ciliated bronchial epithelial cells through an inhalation. But that's it. There's 197 plus cells that of innovation that need to occur. But all of this infusion of capital for Arcturus has made definitely accelerated our early pipeline of research and innovation and screening efforts. So we're gonna be able to apply our technology in different areas, different cell types. If the COVID-19 vaccine works really well, guess what's right behind it? A flu vaccine. And the flu shot has plenty of room for improvement, and messenger RNA could be the solution, and Arcturus could play a role in that, hopefully. And so that's an example. And then in these liver therapeutics, there's dozens of diseases that a messenger RNA, if it gets inside a cell, it makes stuff and it replaces stuff. And all of the diseases in the world, or most of them, are because we're missing something or we have a dysfunctional something in our body. And messenger RNA can just go in there and simply replace it, or whatever's missing or whatever's dysfunctional. And so all of this money that's going into messenger RNA is going to impact all of these diseases, where getting these messenger RNA molecules to the right tissue, to the right cell, to make just a whole variety of awesome proteins that can be, impactful in the pharmaceutical world.
Oh, awesome. So lots of opportunities there. Thank you, Joe. Any other closing remarks by anybody before we wrap up? Or any more questions from the audience, please write it in the chat if there is. Okay, I think we can call it a wrap then. Thank you so much for being on this panel today, and for all the wonderful work that you're doing with the fight against COVID. Look forward to staying in touch, and thanks to all the attendees that came today as well. So,
yes, thank you guys.
Yeah, nice to see you again, Pamela and Joe and Sonia. Great to be on a panel with you.
Nice job, Pam.
Thank you. Thank you. Nice job to all you, to everyone here as well.
Yeah.
Thanks.
Thanks.
Thank you.